So welcome back to my new video. In this video we are going to discuss about some type of white blood cells. In the last class we have discussed about the neutrophils, and we know that neutrophils were the most abundant white blood cells. Their percentage is 50 to 70%. They are short-lived, have a specific receptors on the surface of bacteria by which bacteria are attached. When they become mature, they have the 3 to 5 lobes, and they can migrate from blood to the tissues by maturation, and they secrete enzyme called lysozyme for lysis of the bacterial cell wall, and they are for specifically bacteria. They specifically phagocytize the bacteria. So the monocytes are a type of white blood cells, a type of granulated white blood cells, and they have a bean shaped bean-shaped nucleus. They have the bean-shaped nucleus, and these are uh, 2 to 8%, 2 to 8% of total white blood cells, 2 to 8% of all white blood cells. They are 2 to 8% of white blood cells, and they are originated from uh, myeloid stem cells, and they get maturation. Their maturation occurs in thymus. They are matured in thymus. These are originated from myeloid stem cells, and maturation occurs in thymus. uh, their lifetime in the blood circulation, in blood, they can survive two to they can survive 2 to 5 days. They can survive 2 to 5 days, but they have ability to get to migrate from blood to the tissues, to the lymphoid as well as non-lymphoid tissues. They can migrate from blood into the tissues, and when these monocytes are uh, transferred or migrated from blood to tissues called macrophages. When these monocytes are uh, migrated from blood to the tissues, when these are migrated from blood to tissues, these are called macrophages, because in tissues they grow in size and get more matured, and called macrophages. And these macrophages in the tissues uh, can live months to year months to year. So monocytes are the type of white blood cells, granulated white blood cells. They have non-granulated cytoplasm. 2 to 8% of all white blood cells, they are originated from myeloid stem cells. Maturation in thymus. In blood, they can survive 2 to 5 days, and in blood, these monocytes are the cells called monocytes. When they are matured in the thymus and they can migrate from blood to the tissues, they are called macrophages. And in tissues, they can survive, they can live month to year. So we can write that monocytes in tissues called macrophages and are long lived So this was about the monocytes. Now we are discussing about macrophages. So macrophages are derived from monocytes, derived from they are derived from monocytes and involved in phagocytosis of foreign particles of foreign cells in the tissues. When the foreign particles are involved, these are responsible for the phago phagocytosis of pathogen. We can say pathogens are foreign particles, and these have a large horseshoe nucleus. They have a large horseshoe nucleus, and these are not involved. These are not only involved in the process of phagocytosis of pathogens or the foreign particles. They are also uh, they are also they are also involved in they are also involved in uh, presenting antigens to T cells. Also involved in presenting antigens presenting antigens to T cells. These are also involved in presenting antigens to T cells, and when by this process, by presenting antigens to T cells, these macrophages are also called as ACGS, which means antigen presenting antigen presenting cells. Antigen presenting cells. ACGS means antigen presenting. ACPS, ACP, antigen presenting cells. So macrophages are also called as antigen presenting cells because they are also involved in the process of presenting antigens to T cells, of foreign particles or pathogens, uh, and they release a specific signaling molecules called cytokines. Macrophages also they also release signaling molecules. They also release signaling molecules called cytokines, and cytokines are responsible. The cytokines are responsible for uh, the cytokines are responsible for the activation of other immune cells, and responsible for activation of other immune cells. This is also involved in the activation of other immune cells, and macrophages secrete an enzyme called nitric they secrete an enzyme called nitric oxide for the secret enzyme called nitric oxide, and used to kill the pathogens. Nitric oxide is also used to kill pathogens, and some examples of macrophages include uh, Kupffer cells in liver. These are also macrophages. Uh, Langerhans cells in skins are the example of macrophages, and microglia in central nervous system is an example of macrophages, and alveolar macrophages are the macrophages involved are present in the alveoli of the lungs. So this was all about the macrophages. Again, I'm repeating, macrophages are derived from monocytes. These are uh, responsible for phagocytosis of pathogens, and uh, they have a large horseshoe nucleus, also involved in presenting antigens to T cells of pathogen of phagocytized bacteria or phagocytized pathogens. They are for this process, for this function, they are called as antigen presenting cells, and they also release signaling molecules called cytokines, which is responsible for activation of other immune cells. And nitric oxide, macrophages also secrete an enzyme called nitric oxide used to kill pathogens. Uh, during the encounter, they release an enzyme. They release chemicals. Chemical release release chemical called uh, interleukin 1. This is used to uh, increase in, increase body temperature. So macrophages also release a chemical called interleukin when at the time of encounter, when they are near to the pathogens. When at the time of encounter, they release a chemical called interleukin, which is responsible for the increasing of body temperature. And now we are discussing about the dendritic cells. Dendritic 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 cells. So in the last cell of process phagocytosis, the last white blood cell is dendritic cells. These are also a type of white blood cell. This is also dendritic cells are also a type of white blood cells, and they are used. They are also they also perform the same function as macrophages, which is they present antigens to T cells of phagocytized pathogens. They present antigens to T cells of phagocytized pathogens, and hence called antigen presenting cells. These are also the antigen presenting cells, and known as antigen presenting cells because present antigen to T cells. They are also known as antigen presenting cells because they present antigen to T cells. They are named as dendritic cells because uh, have long cytoplasmic projections on cytoplasmic projection or dendrites. They have long cytoplasmic projections or dendrites, and they are activated by the inflammatory cytokines. Uh, the chemical which was released by the macrophages, chemical which was released by the macrophages, here the releasing molecules, chemical releasing molecules, which is cytokines. These are, here we know that they are used for the activation of other immune cells. So these cytokines are also used for the activation of dendritic cells. Cytokines are used for activation of dendritic cells. For dendritic cells, and these can migrate to the secondary lymphoid tissues such as tonsils, uh, tonsils, spleen, uh, Pear's patch, etc. So dendritic cells are the have long cytoplasmic projections or dendrites. They are known as antigen presenting cells because present antigen to T cells, and these are activated by the cytokines which was secreted by macrophages. Cytokines are responsible for the activation of dendritic cells. They perform the process of phagocytosis by presenting antigens to T cells, which means by activating the T cells. They activate T cells by presenting antigens, and then T cells further uh, work as a immune cell, which they protect our body from the pathogens, from the foreign particles. So this was all about today.